Section 1.4, Part 1. If you haven't done so already, make sure you take a look at the vocabulary on page 74 in your notes and make sure that's up to date. Okay, so final biology topic. We're going to look at GMOs and recombinant DNA. So GMO is short for genetically modified organism. It's an organism or a microorganism, so it doesn't really matter how big it is, whose genetic material has been altered to contain a segment of DNA from another organism. Okay, so its genes have been modified. Modern recombinant DNA technology basically enables the stitching together of pieces of DNA. So it doesn't really matter where the pieces of DNA come from. So remember that all DNA has those same 4 bases. They all have the deoxyribosin phosphate backbone. It looks the same no matter what organism it's coming from, and DNA doesn't know any different, basically. Okay, so this would be an example of some stitched together DNA, overly glorified, obviously. So as scientists learn more about DNA and how it works, they develop the technology to genetically alter an organism's genome. So genetic engineering is altering the genetic makeup of an organism by cutting DNA from one organism and then inserting fragments of that DNA into the genome of another organism. The end result is something called recombinant DNA, which is a fancy way of saying DNA that is made from 2 or more different organisms that have been recombined together. So, for example, glow-in-the-dark plants. So scientists have taken a firefly gene from a firefly. They conveniently named the gene that makes fireflies glow the firefly gene, and then insert it into the plant cell using cut and paste enzymes. So we end up with a plant that glows the same way a firefly would, which is pretty cool. Any organism that has DNA that comes from another organism is considered to be transgenic, where trans means across and then gene means race. In some cases, genetic engineering changes the allele frequency of a population by artificial means, which means that you can end up with an allele showing up more or less frequently than it kind of would normally under natural reproduction. So the steps of genetic engineering. There's basically 3 steps for genetic engineering. I guess this is going back to changing that allele frequency. So if you introduce the glow plant from the above into the wild and it survives for its offspring, you've introduced a new allele into the population. Okay, so 3 steps of genetic engineering. So 1 is isolation. So you have to isolate the gene that you want. 2 is insertion. You have to insert it into the new strand of DNA. And then 3 is transformation. And we'll take a look at what all those steps actually look like. So these steps rely on enzymes, plasmids, and vectors. And we'll talk about all those are as well. So first step, isolation. So the desired DNA segment, so that's one that has the gene that you want, has to be isolated. And it's done by cutting it out of the DNA strand. Okay, so this is showing it with scissors. It's not actually scissors. This would be an enzyme. Okay, so you use some kind of an enzyme, which is a protein, to cut the section of DNA that you want. Okay, and then the enzymes are considered to be restrictive enzymes because they're used to cut the DNA at very specific sites. So it's very restrictive in where it can cut. And just like scissors leave behind kind of jagged edges on like paper or whatever, it's not perfectly smooth. You end up with these sort of jagged, kind of sticky ends of DNA. So this line right here, this is where you would cut. Okay, and because it's cutting between the strands, kind of like between the complementary bases, you end up with these sort of sticky bases. So you end up with these bases that really, really want to attach to something new. So what you end up with is, once again, this sort of cutting taking place. Okay. Along these edges. Okay. And then you have these parts here that really want to bind. Okay. And that's what we call sticky. Okay, so when we say it's a sticky end, it's because it wants to bind to something. You've got these base pairs that are not paired. They're just bases that are waiting to be paired with something different. Okay, so that's isolation, getting out the gene that you want. Then insertion. So next, the DNA segment is put into a vehicle or a vector that will transmit the DNA to the host cell. So a vector is basically just your way of getting it from point A to point B. You can literally think about it as being like a car or a truck or whatever, just on an itty-bitty scale. Usually our vector is a bacterium being the singular for bacteria, or a virus. It could be a pipette, so like a teeny tiny dropper basically, a metal bullet that's covered with DNA. Okay. A common vector in bacteria is called a plasmid, and it's basically a separate little ring of DNA that is present within the bacterium so that it doesn't actually interfere with the bacterium's own DNA. And it's just a separate ring to basically get it into the cell that you want. Okay, so the vectors do sort of dirty work because they actually insert the DNA into the host genome. So you don't want to disrupt the cell with like actually getting in there and cutting, because you don't want to do damage to the cell that you're trying to get the new DNA into. Okay, as the vector with the inserted gene is considered to be transgenic because it is DNA from two different sources. Okay, so here's a viral vector carrying a new gene. Okay, so here's our cell with our original genes. We've got our viral vector with the new gene. Okay, and then it's inserting that one in. Okay, and then that new gene is now present in the cell along with all the original genes. And the third and final step is transformation. So the DNA is then transferred to the host, which ends up being our transgenic organism. Okay, so we've got so bacterial plasmid, okay, with our desired gene kind of getting in, and then it goes into the plasmid, and then that's our bacteria, and then it's plated, and then you kind of get what you want. So an example of this, which I mean probably doesn't exist yet, could potentially, having like a vanilla plant, and you would have restrictive enzymes cutting a gene out, specifically giving vanilla flavor. And then you would have a virus vector bringing that to an embryo of a cow. Okay, so you're starting from like you're not trying to insert this into a grown-up cow. You're inserting it into the first cell of a cow. And then when your cow grows up, it would then potentially make vanilla flavored milk. Wouldn't that be lovely? I think chocolate would be more fun. But yeah, so that is our 3 steps of genetic engineering. There's other examples of transgenic organisms, so these are like real ones, they're not in your notes. So Genie is the first genetically engineered animal to produce a human protein drug, which is human protein C in her milk. We have fish that glow, kind of like our firefly plant. We have goats that can spin webs like spider web. We have transgenic tomatoes, which were the first genetically engineered food that went to the market when the FDA approved the sale of tomato that had been genetically modified to stay riper longer off the vine, so they could be picked picked sort of riper and transported without them getting kind of mushy by the time they actually got sold. And then on the right here, we have chickens that have been bred or have been genetically engineered to not have feathers because it makes them easier to use for slaughter, which is kind of disturbing, but also kind of cool.